Good morning, good afternoon, good evening. This is Dr. DeChavo, and I wanted to start taking a little time to go through the topics themselves. So if we go into, obviously, Module 1, we're going to be looking at the cell, inflammation, repair of tissues, regeneration, and fibrosis. Remember, cells are like little people. Okay, they have to eat, they have to breathe, they have to metabolize, they have to excrete. So the better you understand a cell, the better you can understand pathophysiology, okay? And how tissues will and won't regenerate. You have to keep in mind too, is a tissue able to regenerate or not? So coming down is just a brief overview of path itself, and pathophysiology is understanding the process of the structure and the function of the body, so you can understand and apply that knowledge to disease processes. It's presumed that you have a basic understanding of anatomy and physiology. I understand that that's difficult for some people. It might be a good idea to go back and review some of those structures. Now, Module 1 will look at the cell, how it can be repaired, how it can get damaged. And we're also going to review fluids and electrolytes, acid base, which is very important. That'll be applicable to several of your patients, and how electrical activity can cause a nerve and a muscle to function. We're going to look at that as well. So moving into chapter 1. And once again, I'm not going to go through everything, but I wanted to sort of get the basics down for you. I opened the wrong PowerPoint, pardon me. Okay. My advice to you as you go along is when you get to something you don't understand, stop and look it up. It would print these notes. If you print them with 3 slides per page, you get lines that you can make notes, and it won't blow through your printer ink too. So looking at cells, we have prokaryotes and eukaryotes. Okay, prokaryotes we're not going to deal with. Those are plant cells. We are eukaryotes. You are a eukaryote. It's complex. It's what we will see if you were to scrape off some of your skin cells, or when you do the swab inside a mouth. It will be a cell with a nucleus that can replicate and function on its own. It can move, it can groove, it can secrete, it can bring in things. And by respiration, what that means is bring in oxygen and glucose, just like we bring in oxygen and glucose and make energy, reproduction, etc. Okay. A cellular function that occurs in all cell types. I'm not sure I like this question. Okay, not all cell types reproduce. I think that is saying specific functions, but most cells can secrete in our body. Most cells can move in our body. Most cells can reproduce by mitosis. What it really is looking for is metabolic absorption. So I don't like that question personally. These are not my notes, but they're directly from the book. So I would not give that to you as a question. But I can take this moment and explain that metabolism is a breaking down and the building up of structures. Our body cells metabolize all the time because we've got to bring in substances to repair cells. We've got to excrete wastes to rebuild them, because you got to get rid of stuff if you're repairing. Okay, so I'm going to kind of go over that one quickly. You are eukaryotic. We have eukaryotic cells. It's got a nucleus inside so it can think and function. It's got an outside plasma membrane, it has cytoplasm, and then it has organelles. And you're probably used to that basic structure of this cell. And once again, if you're not understanding something, come right over to this wonderful internet. And images are great for me. This probably rings a bell from A&P. Okay. Sorry, I'm trying to just bring this cell up a little bit. Okay, so there's a picture there, and you've got this cell. This is the plasma membrane. This is the nucleus, and then we have all these structures in here. One of the ones they talk about a lot is mitochondria, and that probably rings a bell because mitochondria create ATP. Okay, ATP are these little packets of energy that you can use in the body. So once again, we'll go back to the notes, but I'm just trying to get you into, how you could kind of work with these notes to learn more. Now, within the cell, you have the nucleus. That helps not only with cell division, but it stores all our genetic information. So did you ever wonder why our genetic code is same from cell to cell, but our cells are so different? I could tell you really quickly, that's because different parts of that genetic code is turned on inside the nucleus. So that huge long genetic code that we have, different parts of it are turned on in different cells. That creates the proteins for that cell that makes that cell function individually. That's why our eye cells are so different from our skin cells, but they all have the same genetic code. Different parts of that genetic code is turned on. Okay. Here's a picture of the nucleus. We're able to get information, some information in and out of the nucleus so we can create proteins and also replicate cells. Here's another picture of the nucleus. Now, cytoplasm is the gunk that holds all the structures in a cell in place. Okay, it's also known as cytosol, or it's made up of cytosol, and it holds cytoplasmic organelles, which are little organs inside of the cells. Okay, and here, there was the picture. Okay, so this is the plasma membrane, this is the nucleus, and then here are all the organelles, which are tiny organs inside of that cell. Now, on the surface of cells, we have different structures that help interact with its environment. We have little protein channels, which can help attach to other cells. We have, this looks like cilia. They're calling it microfilaments, but we have cilia. We see those in respiratory cells. So on the surface, we have different structures, and those structures allow that cell to interact with the external environment. Different organelles. Okay, I'm not going to get a ton into this. I'm going to let you read through those on your own. These are used for making proteins that help with the structure and the function of cells, ribosomes, and endoplasmic reticulum. Golgi complex helps to package proteins to be exported. So we make proteins here, we export them here. I call this the UPS of the cell. And then they can be exported out into the external environment of the cell, proteins to help create structures of the body. Lysosomes help to destroy things that we don't need anymore or are infectious. These do the same thing. Okay, mitochondria. Gosh, I've so much talk about mitochondria, so we will spend a little time on this. I feel like maybe your boards, I don't know anything about your boards, but I'm guessing everybody likes mitochondria. Mitochondria are these little structures that look like kidney beans, and then they have these folded membranes on the inside. And the basic gist of them is to get, ideally, glucose and oxygen into them to create ATP. ATP are little packets of energy that you have in your body. ATP goes out to cells in the body and basically sit there and wait to use energy. And that process is very complicated, that's why I'm not getting into it. But that's a basic gist in it. Okay, so mitochondria, powerhouse of the cell. They have these other structures as well, but I really think you're going to see mitochondria deal with energy. Cytoskeleton, so let's start to think about affiliating words that we already know and using it to our benefit. Cytoskeleton in a eukaryotic cell helps to create structure just like our skeleton does. Okay. Question 2. Function of cellular energy metabolism and generation. Most of cells, ATP occurs in the, let's go over test taking strategies here. Okay, you may have just read through that and be like, holy crap, what does that mean? Okay, so let's go through it. The function. So we're not talking about a structure, we're talking about the function of cellular energy. So we're talking about creating energy. Metabolism is a creation and destruction of energy. Okay, so we're talking about function, cell energy, metabolism, generation. Oh wait, she just talked about ATP. ATP are little energy packets that we create by the powerhouse of the cell, mitochondria. You really want to break every question down that meticulously. Now, if you have no idea what this means, according to what we have said already, the nucleus, I told you, stores the genetic code. It's used for division of a cell and creation of proteins, not energy. Lysosomes destroy structures in the cell. That could be infectious, or it can be old parts of cells that have broken down. Oh, wait a minute. Yeah, she talked about mitochondria. Yes, function of energy. Woohoo. Okay, so see, and then this is packaging of cells. So you want to read every word carefully, but then you also can eliminate by going through the answers as well. Okay, so C, whew, we're right. Now, also you can use these explanations as ways to review what you've learned already and maybe pick up some more tips. Plasma membrane is that outer membrane. It helps not only with the shape of the cell, communicating with other cell, and also movement. It has what's called a lipid bilayer, which makes it what's called selectively permeable. The whole gist with cells, guys, is keeping them at the appropriate size they should be and keeping the appropriate substances inside of them that should be there. It's just like you're hooking somebody up to an IV. You don't want to pump them through full of salt. You don't want to pump them full of distilled water. Think of cells as being little people that you're sticking an IV in, and you want to keep them with isotonic solutions, right? So that is how I want you to think of cells. They have a certain balance they need to maintain. Okay? Plasma membrane has a lipid bilayer. It's actually called a phospholipid bilayer. Don't worry about it. It's also hydrophobic and hydrophilic. And this makes it selectively permeable. So let me look and see if there's a picture first. Yes, there is. Okay, cool. Okay, so we can't get everything into the cell. We can't get everything out of the cell. We wouldn't want to do that, right? We want it to be selectively permeable so we can keep the inside of the cell balanced in an isotonic way. And if you don't remember what isotonic is, shame on you. You should go look it up. Isotonic means that fluids are the same approximate solution as a solution in our bodies. I shouldn't say shame on you, but you should know that. I am so tonic is how I remembered it. Okay? Now, we have to allow certain things into and out of cells. Some things can sneak through that lipid membrane. Some have to go through protein channels. So I'm going to jump ahead here. Okay, so if we take a look here, I'm trying to, this is not what I want. Excuse me for just a second. All right, I'm trying to pull up my drawing tool, which is not working for me right now. Okay, let me draw. Okay, so this is the lipid membrane. Some things can get through it directly, such as oxygen. Okay. Now, otherwise, we have to pump things through these protein channels. So they can either go through them or attach to them. Okay. For example, ions, sodium and potassium will pass through these channels, and they are a big function of balancing solutions, but also the ability for nerves and muscles to fire. We'll get into that in a little bit. Plasma membranes, and you will look at this when you go through pharmacology, can also be stimulated by substances attaching to these proteins and stimulating what's called second messengers or passing through the membrane itself. That's getting above my pay grade. I joke that I don't do drugs, but I don't. Okay, so just understand that in order to get substances through, they have to attach to or go through this plasma membrane. Let me see if I can, oh, I can, great, okay. So protein regulation will be those protein channels that substances can get through, okay? It also helps with the structure is what that is saying. I feel like this is a little wonky here, okay? So ribosomes help to create the proteins in a cell that can come through. We have second messengers, I believe that's what that means, that help to transmit the signal through that membrane. I may be wrong, but I don't believe that's something we need to deal with. Okay, so plasma membrane. We also have carbohydrates that are in that plasma membrane, but I think the big time players really to think about are those phospholipids or those lipids in the protein channels. And the carbohydrates can help with these structures as well, or these functions as well. So, plasma membrane receptors are those protein channels. Okay, ligands are substances that will bind with those protein channels and either turn a cell on or turn a cell off. Okay, and it will really apply when you start to talk about pharmacology, but just to make you aware of that. Okay, so you can have cells adhere to each other. There are different ways we can do that. We have the cell itself, and don't forget that these cell-to-cell adhesions will be, there's the plasma membrane, here's the nucleus. I'm trying to make it a little more solid. Okay, so we're talking about how a cell can contact to another cell. And this will apply in different areas of the body. The best example I can give you is the skin. Okay, you have tightly packed cells on the skin, and they are held together by some molecules and also some junctions between the cells, and also an extracellular membrane. This is a layer, kind of like a sugary layer that's around the outside of this cell that can have them or help them stick to each other. Okay, so cell-to-cell adhesions we'll see in different areas of the body. The best example I can give you is the skin, and it helps to hold the cells together to provide a protective mechanism. We don't, on the skin, if you think of these two cells, 1, 2, as being some of the millions of cells in the skin, you don't want a lot of stuff sneaking in, right? What happens when somebody has a full thickness burn? They lose fluids. You could get fluids leaving. You could get infection coming in. So we want to have the ability to have cells stick together in different abilities. Okay. Extracellular matrix. Okay. That is that slimy layer that's on the outside of the cell. That not only helps them to stick together, that slimy layer on the outside of the cell also helps to hide cells. Cancer cells, this is some of the research that's been done, they actually hide themselves with this slimy layer outside of the cell. Basement membrane helps to secrete that extracellular matrix. So if we're looking at the cell itself, I don't want to get too much into this. I don't think we'll get too crazy with this, but okay. We have this cell itself where it attaches to, let's say this is a skin cell. Okay. The basement membrane is what it sits on. Okay. And it will secrete an extracellular matrix to surround those cells and go between those cells. That's I think about as much as you need to know of that. Okay. And you can see here's a cell. This is called epithelium. This is lining and covering cells. Here's that basement membrane that can secrete that slimy layer. I think that's about what you need to know from here. Okay, cell junctions. I think these are important to know. If we are talking about the cells that line the GI tract, so they are tall and skinny. Okay. And they have, so we're talking about the GI tract, they have to be fairly tight in the GI tract because think about all the bacteria and stuff that you ingest. So tight junctions are kind of little spot welding that hold these GI cells together. So here's Mr. Bacteria that you just ate in your salad today. He's like, oh, can't get through because of these tight junctions. And this is, you've probably heard people who have leaky gut, these get loose and we could get more substances through. Okay, so tight in areas we want to create protection between cells to keep external substances out. Desmosomes, unite cells, are not nearly as tight. We see these in the skin. Now, gap junctions are communications between cells. We see these in the heart cells. Heart cells are Y-shaped cells. Oh boy, I'm going to do my best. Okay. And one heart cell talks to the next heart cell. Okay. And gap junctions are actually tube-like structures that hold the cells together, but because the heart beats so frequently, it allows ions to pass from one cell to the next quickly. So that way it can communicate quickly and allow those substances to flow through. So gap junctions we'll see in cardiac cells, okay? Asymmetrical hemidesmosomes, I've never heard of those before. I'm going to take a chance and say you don't really need to know them, okay? Gating. Gating means that uninjured cells will protect themselves from injured neighbors. It'll shut those communications between cells down, hopefully to stop the spread of diseases and anything that may not be coming from one to the next. Okay, so this shows gap junctions, some desmosomes, and tight junctions. Cell communication. We spoke about that gap junction, okay, sending ions from one heart cell to the next. There's intracellular, which is between the cell receptors, are those protein channels that'll talk to each other. Plasma membrane also has protein channels which send signals across. Let's talk about chemical signaling. Cells in an area can secrete these chemicals that can send an immediate signal. Let's talk about prostaglandins. I think that's a good example. For example, in prostaglandins, once a woman starts to contract, the uterus starts to contract during childbirth, prostaglandins are released from the uterus to signal the other uterine cells to start contracting. Injury. You have an injury in an area, the cells in that area will release prostaglandins. Okay, so chemical signaling will be from the cells itself. And once again, please look up the rest of them so you have a general idea. Because I'm trying to keep these lectures somewhat short, which is probably not going to happen, that signals for those cells to start functioning due to an immediate response. Neurotransmitters will be from a nerve to a receptor, okay? A nerve to a muscle, a nerve to a nerve to an organ, okay? You have several neurotransmitters in different areas. So cell communication. Okay, you can see the different ones that we spoke about. And here's another sign for that. Okay, we spoke about these protein receptors. These protein receptors will get signals. Generally, those are hormones that will activate what are called protein kinases inside of the cell that instructs that cell to do those functions. Okay. Metabolism. So, anabolism builds up, catabolism breaks down. You use energy to build structures, okay? So suppose you are a bodybuilder. You need to use energy to build those muscles. Catabolism breaks down stored structures that we have, such as glycogen, to provide glucose for the body to function. Okay? So anabolism builds. I always remember that because it's like anabolic steroids, right? If you take anabolic steroids, you build up those structures. Catabolism, catabolic, somebody said cannibalism once. I don't know if that breaks it down. ATP are those little packets of energy. Remember that we create them in the mitochondria. The other thing that is important for you to understand In order for mitochondria to create ATP, you need glucose and oxygen. Brief story. My second son never slept. And I would literally, knowing that ATP is created in a cell in the mitochondria with glucose and oxygen, would literally sit while I was feeding him or nursing him and eat a power bar with glucose and breathe and think, okay, I gotta make some ATP here, you know. So that is the best environment when you have glucose and oxygen to create energy. We can create energy from fat. It's not as efficient as glucose. We can create energy from protein. That is really not good. That means we are getting into proteins, the structure of the body. We're breaking down the structure of our body to create energy. And that happens with severe anorexia or wasting. Okay, so glucose, which is a carbohydrate, is the best thing we could use to create ATP. Lipids or fats are the second choice. Protein, we're getting into breaking down the structures of the body. Okay. We digest proteins, fats, and carbohydrates. Carbohydrates include polysaccharides. Generally, people call it, when you're talking about those large structures, carbs are breads, pastas. Proteins are our meats or our soy if you're a vegetarian. Fats are those lipids that we bring in, oils, etc. We need all of them for different functions of the body. Carbs are generally used, and I would add carbs right here. Polysaccharides are one of the carbs. Sorry, cannot. Carbs. Oh, I apologize. C-A-R-B. Okay, so carbs. Polysaccharide is a carb. So carbs are used to create energy. Okay, and proteins are used to create the structures and the muscles of the body. Fats are used as a secondary source of energy. Proteins are also used as hormones. Fats are also used as hormones. Fats create the structure and the plasma membrane of cells. So those all have important functions. Glycolysis is the breakdown of glycogen into glucose units. So we have these long strands of glycogen. Let me show you a picture. Okay, so glycogen. Oops, hold on. Let me see if I can just pop in here. Glycogen. Okay, glycogen is stored. Cool picture. Alert, nerd alert, nerd alert. Okay, so each one of these is a glucose unit. And what happens in our liver and our muscles is we have glycogen, which are long chains of glucose. When we don't eat for a long period of time, we start cleaving these off for energy. Now, we also have this stored in muscles, but muscles like to use it for themselves. They're very selfish. So the liver is the primary area that we store glycogen, and we store it for the future use of glucose. Okay? Okay, so glycolysis is the breaking down of those glycogen chains into glucose units so we can use them to produce ATP in the mitochondria. Okay. We produce it in what's called the citric acid cycle. So we'll feed it in, it's turned into another structure, I'll tell you in just a second. Goes through the citric acid cycle, also called the Krebs cycle, and we produce a whole bunch of ATP. Okay, so here we go. We feed it in, okay, to this step of glycolysis. That was what they were talking about previously. How they have it broken down. Okay, we then turn glucose into pyruvate and acetyl coenzyme A. I think I used to know every single step of this. I'm going to try to keep it simple for you guys. It goes through what's called the citric acid cycle, and then ATP is created through what's called oxidative phosphorylation. Okay. I'm really trying to pare this down for you, and I'm hoping it's as simple as this. So glycolysis is going to be, taking that food, whittling it down to acetyl coenzyme A. That is then fed into the citric acid cycle, which will then go through oxidative phosphorylation where ATP is made. Okay, I'm trying to keep this super simple. And all of this happens in and around mitochondria. So we bring in glucose, we eat it, num num num. We breathe oxygen. Okay, oxygen and glucose go into this process, goes through those few steps that I tried to narrow it down for you and create a crap ton of ATP. That ATP sits around and waits until we need energy. That's a basic gist of it. Okay. Okay. So oxidative phosphorylation is where we actually make the ATP. Now we need NAD, which actually comes from B vitamins. Okay, that may ring a bell. If you don't have those B vitamins and that's missing, you don't create as much energy. Okay. And please realize I'm super simplifying this for you. So if you know all of those steps, that's awesome. But I'm trying to really break it down. Now, okay, so if we do not have oxygen, can't run the whole process, okay? It'll just stop at pyruvic acid. You get 2 ATP, that's it. That's why if you sprint really fast, you squeeze those muscles, you can't get blood into them, you get really stiff the next day because you've gone through anaerobic glycolysis, you've created lactic acid as a byproduct. Okay? When we bring oxygen back, it goes back to this oxidative phosphorylation. Yay, we have energy. Okay? Here's a chart if that helps you. I've tried to break it down as best as I can for you. Okay, so membrane transport, we've kind of gone through this. We need to bring structures or substances in and out. We can have a few things sneak through that lipid membrane, but for the most part, it's got to go through those protein channels we talked about. We can do this passively, we can do this actively. That simply means we can either use energy or not use energy. So with active transport, some of these pumps use energy, and don't forget energy is in little packets called ATP. There are 3 main transport systems that will require that energy, and I believe it is, there's a sodium potassium pump, should be here. Okay. Endocytosis and exocytosis use active energy. Osmosis, diffusion, and hydrostatic pressure do not use energy. So let's look down there. Okay. So media transport will go across those channels either together, opposite each other, okay, or a single molecule moved in one direction. I think this is best shown on this picture. So here's our little protein channels. Uniport, somebody's just sneaking on through. That's all that means. Okay. This one, symport, means that these two guys are meant to be together. Isn't that a dry little heart? Oh, okay. They're in love. They're going together. They transport it together. Generally, it has to do with charges that have to go across that protein channel. Antiport means they go opposite each other. For some reason, in order to balance this cell out, we need to move one in and one out. Okay. Electrolytes, about 95% of the solutes in body fluids. Okay, so a solute, if you don't remember, if this is a solution of salt water, water is the solvent, yes, and the and then the salt, I'm sorry, I just totally went blank there, I'm not gonna lie. Okay, so if we're talking about a salt water solution, my apologies, solvent is water, solute is salt. So the solute is the stuff that's dissolved in the solvent. We don't talk about a ton of those. We're going to get to your basics, you know, like your glucose, your sodium, your potassium. Okay. So electrolytes primarily are going to be sodium and potassium that we're going to talk about. They're measured in milliequivalents per liter, per deciliter. Okay. If you need to know that. Cation will be positive and anion will be negative. Okay, so just to get a general gist. So we will be focusing on the solution that's inside and outside of the cell. Okay, the solution inside of the cell is the cytosol, don't forget. And then the solution outside is a solution between the cells in the body. So we can have substances just kind of float in and out passively. If we're talking about the cell membrane, sodium flows in, potassium flows out, okay, and they'll go through those protein channels. It just happens. It's passive. There's no energy. It just happens, okay? So filtration will be movement of water and solutes through a membrane. Really, I think we'll talk about this more when we start talk about perfusion. So when we start to talk about, and I'm sure you're all aware of perfusion and how important it is, I think right now we're going to focus on diffusion, osmosis, and that. Okay, we'll talk about filtration more with perfusion as we move ahead. Okay. Osmosis is the movement of water down a concentration gradient. So okay, let me try to simplify this for you. If we have, and I'm not sure if you can see the little dots I'm making, suppose this is sodium inside of a cell, okay? And this is sodium outside of a cell. So I put a bunch of dots on the inside. I have 3 dots on the outside. Just intuitively speaking, sodium is going to go and try to balance things out, right? Okay? In the same respect, water is going to start to flow in. The whole gist is inside and outside of the cells, we are going to try to balance solutions for the most part. There are a few exceptions. And we're just talking about a resting cell. So osmosis is the movement of water into an area that has a higher solute concentration. And diffusion is the movement of those particles away. It will move both ways. Not this, fluid will move in, solutes will move out to balance those solutions because we're going to try to balance them inside and outside the cells. So osmosis is the movement of the water. Diffusion is the movement of the particles, is the general gist of this whole thing, okay? Okay, you can review that on your own if you like. The balance of those solutions. Okay, I would somewhat memorize those. Active transport is going to use energy. It's going to use ATP. Okay, we have something called a sodium-potassium pump. We also have do we have this? Endoexocytosis. Okay, so sodium-potassium pump will balance sodium and potassium in a cell. Okay, we have in general more potassium inside of a cell and more sodium outside of a cell. But they like to sneak, okay? And they're both positively charged. They like to sneak. He'll sneak in, you'll sneak out. This transport system that uses ATP puts them back where they should belong. I think that's good for now. It's actually way more complicated than that, okay? So active transport, sodium potassium pump, when they sneak in and out of the cell, they get sent back where they should be. Okay. Let's review this. Active mediated transport involves. So transport, think about what we've been talking about across the cell, okay? Movement driven by diffusion, energy, expenditure, moving water. We're talking about active. What did we talk about? Mechanical force of water. Active. Active we've talked about is using ATP. Okay, if we're using ATP, what do you think? I would think this. This sounds like they're using expenditure of metabolic energy. Let's see if we're correct. B. Okay, so some of the questions are tricky like that, I think. So you've got to go through and look for those key terms. Active, energy use, metabolic, expenditure. Okay, that's what they're getting at. And you can review those. I would review, I feel like some of the test questions may come from these answers. So I would review those carefully. Okay, so active transport uses energy with endo and exocytosis. I wonder if they have a picture of that. They do. Okay. Endo, don't forget like endoscopy, in, bringing it in. Vesicle moves into a cell, it ingests it. One of the big ones you may remember this is phagocytosis. That's using cells to eat up things that we don't want anymore or are infectious. Pinocytosis is bringing in fluids. Exocytosis is spitting stuff out. That's how I always remembered it. Stuff that we either don't need in the cell anymore or protein structures that we have started to create that need to go out into the body. Okay? And what happens is structures are brought near the cell, kind of push into the cell, and then that cell membrane closes off, and this now has a nice lipid membrane around it, which is pretty cool. Exocytosis, it's brought to the cell surface, pushes through that membrane. It then has a lipid membrane around it, and it gets on out of there. Okay. All right. Electrical impulses. So this will include what is called an action potential. Now, an action potential is what makes muscles and nerves work. An action potential is an electrical signal. So suppose we have a brain cell for motor to your biceps. Okay, here it is. Here's the nucleus. This is the cell body. You probably remember an axon. Okay. What happens is an action potential goes down that axon and sends a signal to your bicep muscle. I can't draw. I'm so sorry. I can't draw to begin with, let alone with this system. Okay. So an action potential is an electrical signal that's sent down the axon that goes to the muscle and says, hey, you got to move. And then down this muscle, an action potential happens that actually contracts the biceps. Okay. So an action potential, if you were to chart it, actually almost looks like an EEG reading. Okay, interestingly enough. And this is the beginning of it. And when it fires, and this is when the action potential resets itself. And the firing happens by a movement of electrons, just like we have calcium movement that moves through heart muscle cells that gives us that EKG reading. So this movement of electrons creates depolarization, repolarization in the refractory period. This is the depolarization where it fires. This is repolarization where it resets. And this is the refractory period where it hyperpolarizes and resets itself back to normal. And this would be the electrical charting of a resting cell. Okay. So action potential makes nerves and muscles function. Okay. When you chart it, so electrically speaking, if I were to put electrodes in here and chart it, it looks almost like an EKG, but it's not. Completely different. Just trying to get you reset. Depolarizes, repolarizes, and then resets itself during the refractory period. Okay. Hyperpolarization is when it goes below resting. Okay. And hypopolarized is when it goes above. Yeah. All right. I think that's a good start on that one. Okay. Here's a little picture of it. These all happen because of the movement of sodium and potassium ions. And that's getting, I think, above what we need to study right now. They gave you a general gist of it. If you want to know more, I'm happy to help you with that. Cell cycle. Okay. Mitosis is when the cell replicates its DNA. Cytokinesis is when the cell splits, and it has different phases. Okay. Now, so mitosis is when the division of the cell happens, and during these phases is when DNA replicates. Just to give you an idea. Okay, so let's talk about the different phases Sorry, let me just see what else they have in these notes. Okay, so what happens when cells replicate themselves? So we have one cell hanging out, and it's got its strand of DNA. It decides it wants to create new cells. It will create a replication of its strand of DNA. So imagine this is exactly the same. Now, we can't keep the cell like that. It has double the amount of chromosomes, right? And chromatin, which is the genetic material, we now have to split it, okay? During prophase, we form chromosomes. I call it prochromo. C-H-R-O. Oh boy, I bet I could type on these. That might be helpful. Prochromo. Yeah, keep laughing at me. Okay, so you actually get those X-shaped chromosomes. Okay, me on the equator. That's how I always remembered it. Those chromosomes then form on two sides. Okay, and they start to get ready to split. Excuse me. So we've got the genetic material now on two sides of this cell, and they split. Anaphase. They start to split and divide into two different cells. Okay. So the genetic material really goes to the opposite side of the cells, and they start to split. And then telophase is when all the cytoplasm is pinched off. So I remember that by tell the cell to split, and you actually form two cells at that point. Let me get you just a quick to get you a better. Here we go. Okay. Prochroma, you form your chromosomes. Me on the equator, you start to line all the chromosomes up to be split. And there's actual fibers that connect to the chromosomes. And you can see then they start to pull the chromosomes, and they split. They pull them to the opposite sides and then tell the actual cell to split. That way you get replicated cells with the appropriate amount of chromosomes in them. Okay, so if that doesn't make sense, please let me know. Okay. So cell division and growth, things build up, they break down. I'm sure you've all heard that you have a brand new liver every 7 years, right? We need to be able to, in some organs, grow more cells, replicate them, and destroy old ones, okay? Some cells don't have this. As you know, in the central nervous system, we don't have that ability to create new cells. There's some new theories about that, but that's what we're going to stick with for now. This is why if somebody has nerve damage in the central nervous system, it is permanent. Okay, so different tissues have different abilities to create new cells and also to fix their cells. Okay. Tissue formation. A tissue is created of similar cells. They will have different ability to connect to each other. Just as we spoke earlier about cardiac cells having gap junctions and having tight junctions in our GI tract, okay? Different cells have different attributes is what I'm looking for, okay? And that is when I spoke about different cells being different from each other, we have, okay, that specialized patterns of gene expression. If I take a heart cell and I take an eye cell, they're completely different. They have the same DNA. Different parts of that DNA is turned on. That's what I was talking about earlier. Okay, terminally differentiated cells. Got it. Okay, so cells as we start as a newborn, a sperm and an egg cell join together. It forms a blastocyst, which is a bundle of cells. That baby then starts to undergo differentiation of those cells. And it's magical to me, really, to think about that. You know, you form different layers, and from different layers, you get different organs. You know, heart cells turn on a different part of the genetic expression compared to skin cells to eye cells. It's unbelievable. It all just happens. And stem cells are cells that don't have that part of the genetic code turned on, so they can be turned into anything, which is, you know, partly why they are used to experiment with. You know, they used to be taken from, oh God, I hate to even say it, aborted fetuses, but we're finding stem cells in the body now, as you know, and we can use them from bone marrow. There are some stem cells that are in the nasal cavity, which is interesting, right where we smell, where the olfactory nerves are. It's kind of cool. Okay, so different types of cells, different abilities to regenerate. Okay, nerve cells, very limited ability to regenerate, although peripheral nervous tissue can repair. Epithelial, very regenerative. Epithelial is what covers our skin, lining of the respiratory tract, the GI tract. Connective tissue is obviously what connects, yay, something that makes sense. There's tons of different types of connective tissue though. And then muscles, which enables movement, has limited capability to repair. Okay, case studies, I'm going to let you read through those and review those questions on your own. All right. So please let me know if you have any questions about this. Hopefully this helps a little bit. Look forward to talking to you soon. Thanks.